I am now joined by one of the state-of-the-art speakers here at ATC 2023, Dr. Jay Fishman. Doctor, thank you so much for joining me today.
Thanks for having me.
Absolutely. So let's go ahead and dive right in. The title of your presentation is Infection and Transplantation, What Have We Learned from Pigs? So can you tell us, what have we learned from pigs?
So the topic of my talk was really to reintroduce infectious disease as a complication of transplantation in our immunosuppressed patients to the general transplant audience. We have experience going back now over 20 years using miniature swine as organ donors for studies originally in primates and now going into clinical trials in people. And so it's very exciting, a way of rethinking about the risk of infection when you start using another species as your organ donors.
Transplant infectious disease has really emerged as a crucial aspect of transplant care. Could you elaborate on how the field has evolved over the years and the impact it had on improving patient outcomes?
So when I started in this field, which was a long time ago, we didn't have very many good diagnostic tools and we didn't have very many good therapies. And so as we have evolved with the advent of immunology and molecular biology in particular, we've learned a lot about infectious disease, and we've learned how to prevent infectious disease. And so one of my contributions has been to develop a timeline, if you will, of what we can expect in terms of complications after transplantation, and then to develop strategies to prevent those infections so that people can get organ transplants and live the lives that they're supposed to in healthy and productive ways.
Wow, that's incredible to hear. There's been so much evolution throughout the past few years.
It's been very exciting. It's been fun, and you deal with a lot of scientists and young people and you continue to learn for a very long period of time. So it's been an exciting 40-year journey, really.
Standard immunosuppression management has helped in predicting and managing infections in transplant patients. Can you discuss some of the preventative and diagnostic tools that have been developed as a result of this approach?
So when I started, our immunosuppression, the drugs we used to prevent the body from rejecting various organs, was pretty crude. And as the immunosuppression has gotten better, we tend to use standard regimens for organ immunosuppression across the entire world. And that has allowed us to look at a timeline and to predict that, for example, we need 3 months of prevention against cytomegalovirus, or we need 6 months of protection against pneumocystis, a common respiratory pathogen. So that with that advent, what we've been able to do is protect our patients from the most common infections and move on to using new or better immunosuppression with less risk. Again, it's all about getting the people back into the community and to be safe in doing things they want. But it used to be that if you took up gardening or you took up some sort of animal breeding or whatever you did after you transplant, you'd get sick from your exposures. Now we can predict that, and now we can prevent that with greater certainty.
The field of transplantation faces a lot of new challenges, such as xenotransplantation. How do you see these challenges potentially altering the current paradigms of transplant care, and what are the associated risks and the potential benefits?
So one of the things about anything that's dramatically new is it makes you look at what you've been doing for a long period of time with a critical eye. So in xenotransplantation, the goal is to have enough organs so that we can provide organs for everybody who needs them, which we can't right now. We have a shortage. With that in mind, and with the new technologies available, particularly genetic manipulations, we've been able to develop pigs as organ donors for people. That means we've applied, and you've probably heard about CRISPR, we've heard of various genetic manipulations that have allowed us to make xenotransplantation a possibility. Then what we do is we say, how do we get from here to the next step? And one of the next steps is safe pigs that don't have organisms in them that will infect humans. And so we develop diagnostic and prophylactic strategies for those. So it's very exciting, but it makes us look back at what we've been doing all along, thinking that we knew so much and finding out we didn't know nearly as much as we thought we did. So it's re-education of us as well as the community, because the person in the community who's not in the field has to be able to look at this and say, this is acceptable to me, that it's okay if I get a heart from a pig, or it's okay if I get a kidney from a pig. And that's very exciting. And we're at a meeting where we all get together, and of course that was held off a little bit by COVID, but where we all get together and we exchange ideas and establish new collaborations and try to move the field forward.
Absolutely, that's what it's all about. Balancing the risks and benefits is crucial in transplantation. In your opinion, how can the field of transplant infectious disease contribute to finding the right balance between minimizing risks such as infection while maximizing the benefits of transplantation for patients?
So the goal is to have our programs data-driven. And so as we accumulate more clinical experience, we find out the things that we're not doing well and we correct them. So it's small tweaks on regimens to protect our patients along the way. That means we need to do some new things. We need to look at the immunosuppressive drugs we're using, which have side effects, and think about ways of avoiding using some of those drugs. We have a thing called immune tolerance, where our patients will be on no immunosuppressive drugs. If that's true, their risk of infection will also go away. So we have to have one eye on the future while we're gradually tweaking what we're doing on a day-to-day basis. I think we're all on the same page in terms of making transplantation as safe as possible for our patients or for the community. And as long as we continue to learn and do experiments and do new science, I think that'll happen.
Absolutely. It sounds like there's been a lot of success in the transplantation industry and only more to come.
And great corporate partners and all kinds of great science that we're going to advance the cause of transplantation.
So, Doctor, can you tell us, how can we use modern scientific technology to really advance transplantation?
One of the things we found out is that if we understand the basic mechanisms of disease, that we can devise better diagnostic and therapeutic strategies in the clinic. And so as new technologies become available, new culture systems, new genetic technologies, new immunologic systems, we're able to apply those, figure out how diseases work, and then come back to the patients and say, I can do a better job. So we have to stay on top of the basic science to really advance transplantation globally.
Well, thank you so much for your time. I really appreciate it.
Thanks for talking with me.